Welcome. Today we're going to be discussing a review of the work that you did last week on incomplete and codominance. First thing that I wanted to go over was what the difference was between codominance and incomplete dominance. I was reading over your answers to the question that I assigned in this practice, and it seemed like we needed some clarity. So the big difference between codominance and incomplete dominance is that although in both cases you are going to have three different phenotypes, meaning that you're going to have three different ways in which the organism can look, in codominance, you are going to see both. So you see both alleles equally. But the difference then in incomplete dominance is that you see a mix of the two alleles. So mix for incomplete, you see both equally in codominance. So if you were to have two different alleles, and let's say they were black and white, if these were your alleles, black and white, if it followed incomplete dominance and you had a black and a white allele, this would be gray. But for codominance, if you had a black and white allele, this phenotype would look, let's say, striped. So it would be stripes of black and stripes of white, where in this case they blend together for incomplete. Okay, so first sample problem here for codominance. In certain fish, blue scales and red scales are codominant. When the fish has a hybrid genotype, it has a patchwork of blue and red scales. So keyword here, we have hybrid. Hybrid means the same thing as heterozygous. So if they were to be a hybrid, 1B, 1R, then they have that patchwork phenotype. So the first question, it says, what is the genotype for the blue fish? Well, it's given to you here. So the genotype, you could only have blue alleles, because if you had blue and red, then you have a patchwork. So genotype are my letters. So here it's BB. Genotype for red, it's RR. And genotype for the patchwork, it's BR. So they're not blended together. It's not purple, but it is a patchwork or kind of stripes that intersect of blue and red. Next question, same phenotype that we're working with. It says, what happens if you breed a patchwork fish and a fish that only has blue scales? Well, here I'm going to set up my cross. So my parents' genes, if I have a patchwork fish and a blue scale fish, patchwork is BR, blue scales is BB. So I'm going to put them into my cross. I'll put the patchwork at the top, put the blue on the side, and fill it in. It says, what is the probability of having a fish with red scales? Well, red has to be RR, and none of them have red scales. So that's going to be 0% or 0 out of 4. What is the probability of having a fish with patchwork scales? Patchwork is BR, and I have 1, 2 that have patchwork. So 2 out of 4, or 50%. So incomplete dominance, remember it's going to be different. Here in the phenotype that is a hybrid, you are going to show a blend of the two alleles. So here, if I have snapdragons, it's a flower controlled by incomplete dominance. The two alleles are red and white. The heterozygous genotype is expressed as pink. When I do both co and incomplete dominance, I like to use two different alleles, both with capital letters, because neither of them can hide the other. They're both shown, and that's how I like to set up my problems. So what is the phenotype of a plant that has only R alleles? So 2R Two R alleles. R is red, so this one is going to be red. B, what is the phenotype of a plant with the genotype RW? If they have an R, red, and white allele, well, for incomplete, they're going to mix together. So mixing red and white, I am going to get pink. And then C, what is the phenotype of a plant with the genotype WW? WW, just two white alleles, then the phenotype is going to have to be white. And then the problem here, a pink flower is crossed with a white flower plant. So pink and white. What is the probability of producing a pink flowered plant? Well, pink has to be one red and one white mixing together, and white is just going to be white alleles. I'm going to set that up into my Punnett square. Put the pink at the top, white on the side, and filling it in. That's what I get. Back to the question, it says, what is the probability of producing a pink flowered plant? Well, pink is RW. 2 out of the 4 boxes are pink. So my probability is going to be 50% that will be pink. Okay, a few more problems for you. So what cross will produce the most pink flowered plants? A lot of you struggled with this question. I'd say about half of you. So if I'm going to do a cross that has the most pink flowered plants, I would probably want a cross that's only going to give me pink flowered plants. If I do this first cross, I'm going to have a white allele and a red allele in both of my parents. So both of my parents are going to start as pink. And that might seem like the choice to do, but if I were to actually complete this cross and have both of my parents being pink, then I actually only end up 2 of the 4 boxes that are going to be 50%. So in that case, it's only 50%. But look at this one. If I have one parent that can only give a red allele and one parent that can only give a white allele, that might give me my answer. So let's put it into the Punnett square. Look, every single one of these boxes now is going to give me a pink offspring. So the one that's going to produce the most pink flowered is going to be this one, a cross between a pure red and a pure white. Now the last concept, this is not incomplete dominance. This one is codominance, and this one is also multiple allele. This is your human blood type. So for human blood typing, we have 3 different alleles. A and B are both dominant. In fact, they're codominant, and O is recessive. So I wanted to go over this with you. This is another section where most of you did okay, but there were some questions I think that you might have. So of this shows you the phenotype, what blood type you actually have. So what are the physical protein receptors? The genotype, what are the genes then that will code to make this protein? And then this case, it's who can donate to and who can receive blood from. So depending on the protein receptors that you have, you can only donate or receive blood from somebody that has like receptors, or that they have a protein that you don't make an antibody for. All right, so the question says, write the genotype for each person based on the description above. So here's my genotypes, and there I'm going to give you a phenotype. In some cases, I might give you a little bit more information. So homozygous. Homozygous, remember, means that they only have the same type of allele. So homozygous for B, I have 2 choices for type B blood. The homozygous one is going to be this one. So this one is going to be this. Heterozygous for A. Well, A, I've got 2 choices, but only this one is heterozygous because heterozygous means 2 different. So it's going to be AO. I also like to write my O's as lowercase because O is true recessive here in this case. If you have type O blood, there is only one genotype that you can have. Because O is the recessive phenotype, can only be accomplished by the homozygous recessive genotype. Type A and had a type O parent. Well, I have two choices again, but each parent has to give one allele. So it has to be this one because they would have had a parent that has type O blood, and type O can only have O alleles. Type AB blood, go back to my chart, genotype, only one genotype, has to be AB. Blood can be donated to anybody, okay? I'm looking here, universal donor. In this case, then, it's going to have to be O. O can donate to anybody. And then lastly, can only get blood from type O donor. If I look here, can receive from, it's only going to be this one here. So O can only receive from a type O donor. All right, I hope that helps. If you have any questions, please let me know. Have a good day.